What spice? What twice? Looks like the momentum for DNA is strong, cuties, so let's take it further for a few more episodes. I'll take you on a style venture with cutting art, state-of-the-edge editing programs from 2015. Last time we covered replication, but for now I'd like to shimmy shuffle back a step to review the structure of DNA itself, to review its grooves before we go back to study its moves. Boodle doodle 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 doodle. Won't have to worry about cease and desist from Universal. I'm the only long-bodied mascot for this channel. DNA is arguably one of the most recognizable biological molecules. It's both enigmatic and universally recognizable, making it perfect for inserting into pop culture as a symbol of time to get science-y, or time to get nifty kind of symbolism, or even time for time-traveling dolphin shenanigans. Firstly, the sweet double helix. Sure, it does look twicely fresh, but it begs the question, why come twisty styles? Why come double helix? Well, the double helix is a surprisingly sturdy structure, especially when it comes to the phosphate backbones that are naturally negatively charged. This charge not only helps in structural integrity, but it allows other important molecules to bind to itself for processes such as replication. The double helix shape also allows for effective unwinding and insertion for replication to initiate its oh-so-vital process, as covered in the previous video. You know how objects that combine strength and flexibility tend to be the most resistant to breaking, such as spider silk? Well, though DNA isn't nearly as strong, the double helix does allow it a relatively decent amount of durability against physical threats, such as an experiment Frederick Griffith conducted in the late 20s shows. He had many bacteria heated until death. The bacteria's DNA remained largely intact. Another primary function of the double helix is its ability to be stored so efficiently, a simple but incredibly vital feature. The next feature is pretty groovy. We can't talk about the double helix without talking about its grooves. DNA isn't shaped like two strings wound around each other, but rather a gently twisted ribbon. Because of this unusual but pretty shape, there is a naturally occurring minor groove and major groove. One being much larger and obvious, and the other one feels more uncomfortable around Jared Fogle. Some binding molecules are granted easier access to DNA binding because of the existence of the major groove, giving them more physical space to reach the base pairs housed within. Mentioned in my previous video, we discussed how DNA molecules have directionality, with a unique shape granted by two differing grooves. The directionality is given a reference. When viewed from the 5' to 3' direction, the minor groove gives the DNA a clockwise spiral. The base pairs. These molecules are sequences of information encoded in the form of four primary base pairs. Guanine, represented as G, thymine as T, cytosine as C, and adenine as A. When paired up, A binds with T and C binds with G. You know, just like real life. Ta! And the AT connection is made of a double hydrogen bond, while G and C connections have a triple hydrogen bond, because those two are paranoid and like to make extra work for poor old helicase. Ooh, those backbones! Living up to their titles, the sugar phosphate backbones are the key agents in keeping DNA structural integrity. These long lads house an inherent negative charge, allowing for more efficient binding and structural shaping, keeping the whole deal from becoming a big old pile of spaghetti. The backbones themselves house the base pairs in their respective sequences, each paired not unlike rungs in a ladder. The backbones themselves are made of alternating molecules of sugar groups, of ribose and phosphate groups. As a bit of a side, I feel it's best to show you cuties just how DNA is packed so dang effectively within a little cell. That is definitely worth an episode or two of its own, but it's worth lightly dipping our terminal eggs into. You wanna know about chromosomes? You gotta know about acids. You wanna know about acids? You gotta listen to me. We got nucleodromes, chromodragus, crangos, twisto, swirltopia, and this one's too far away. No name for it yet. You could name it after my mom, if you want. It's up to you. We got the DNA helix itself. That wraps around a spherical molecule known as a histone. The histones organize into loops to form chromonema fibers via molecules known as linker histones. Chromonema fibers eventually form compact loops known as, and bear with me because it's hard to recall, chromatin loops. These goofs wind even more so to form the more recognizable chromosome. Chromosomes on their own would merit a whole video, or 3. Alright guys, share a share and link a like for my video piles. It's all swear-a-cuss free, so even grandma can enjoy the miles of educational styles.